Today, I'm going to be talking about a recently developed method for integrating transcriptome-wide association studies and colocalization analysis. So here's a little bit of a roadmap of today's talk. So first, I'll start with some background, just what is a colocalization analysis? What is a transcriptome-wide association study? Then I'll go into our methodology, which we're calling INTACT and INTACT-GSE. I'll show how it works in a simulation study scenario and a real data scenario, and then finally I'll introduce the software package for this methodology. Okay, so getting into some background. So by now we've had a lot of genome-wide association studies, and these have identified lots of different genetic associations underlying complex traits. But one of the most, one open problem that still stands in statistical genetics is linking these genetic associations to the genes that they target. So we're calling these putative causal genes, or PCGs. So traditionally, this sort of path relies on biological knowledge of genes that are close to the GWAS loci. So for example, in this Manhattan plot here, you can see that the top GWAS hits have all been annotated based on previous biological knowledge. Okay, so recently a new class of methods has begun to emerge that essentially integrates different types of omics data in order to identify PCGs. So the advantage of this sort of approach is it gives it paints a better picture of the underlying biological mechanism for complex traits. So we're calling these mechanism-aware PCG implication methods. And some examples of these types of methods are colocalization analysis and transcriptome-wide association studies, or TWASs. And one of the most common multi-omics data platforms that's used in these types of analyses is gene expression. Okay, so what is a colocalization analysis? The goal of this type of analysis is to determine whether genetic variants that are causal for a molecular phenotype overlap with variants that are causal for a complex trait. So here we have a little cartoon of a simple example, just to show what a colocalized variant might look like. So you see from the cartoon, SNP-X is not only causal for the expression of some gene A, but also for your complex trait. So we say SNP-X represents a colocalized variant. So there are challenges that go along with this type of analysis, one of them being the presence of linkage disequilibrium, and also if there's multiple causal variants at a locus. So this can be a challenge because in identifying SNPs like SNP-X, you might not be sure if the SNP is like SNP-X, or if basically you have individual causal EQTL and an individual causal GWAS hit that are just correlated with one another. All right, so here are some more specifics on what goes in and comes out of a colocalization analysis. So for input, you'll often have something like probabilistic QTL annotations from a fine mapping method, as well as posterior probabilities from your GWAS. And as output, you'll often have what's called a gene level colocalization probability. And this is calculated as 1 minus the product over your signal clusters of 1 minus the RCP. Where the RCP is the probability that a genomic region contains a single colocalized variant. Okay, so what does all that really look like? So again, we have another cartoon, another toy example, just to illustrate how you calculate that gene level colocalization probability. So again, we're returning to gene A. So here you have 3 different signal clusters, R1 through R3, each with a corresponding RCP. And then when we apply our formula, we get 0.995. Which is essentially saying there's a high probability of at least one colocalized variant for this gene. Okay, so moving on from colocalization analysis. So what is a TWAS? So similarly, the goal here is to sort of link our genes to our complex trait, but this time we're essentially evaluating an association between the expression of a gene in the transcriptome and the complex trait of interest. So often how these start out is you have a reference panel, for example, GTEx, if you're looking at expression data that you use to train your expression weights in this cohort. Then you use those weights to predict expression in a separate GWAS cohort. And then you predict that, or you predict expression and then associate that with your GWAS trait. So what you end up with is essentially a transcriptome-wide set of z-scores. And why this sort of analysis is particularly advantageous is because you often don't have data sets that contain both your molecular phenotype data as well as your GWAS trait. So this allows you to sort of reap the benefits of the large sample size that you usually have with a GWAS. Okay, so just a little bit of a summary of what we've just gone over. So colocalization analysis, we're probabilistically quantifying the overlap between causal QTLs and GWAS hits. The output is your gene level colocalization probability. Meanwhile, for TWAS, we're testing association between predicted gene expression and a complex trait, and we're getting a z-score as output. So recent work by Haku et al. Has shown that when these two types of analysis are applied to the same data, they can actually be complementary. So it turns out that they actually don't always implicate the same set of genes. And what this work showed, their basically interesting finding was that this can actually point to certain things going on in the underlying biology. So for example, if you have a gene that has a strong colocalization signal but weak TWAS signal, that can indicate the presence of horizontal pleiotropy. Meanwhile, if sort of the opposite is true, that can indicate the presence of an LV hitchhiking effect. So down here, we have a cartoon to just show the difference between horizontal pleiotropy and vertical pleiotropy. So basically, we want to filter out those cases that might indicate horizontal pleiotropy because we don't have any causality really going on between the gene expression and the complex trait. So sort of the punchline here is we want genes to both have a strong colocalization signal and TWAS signal. Okay, so what did I mean by LV hitchhiking effect? So that's sort of basically this figure tries to explain that. This is from a different paper. So here we have a Manhattan plot showing your GWAS signals. Here we have an expression weight matrix for 6 different genes, genes 1 through G3. Gene 6. And then we have a LD matrix below. So basically what I want to highlight here is gene 3 is the only causal gene, but you can see there's no overlap actually between the gene 3 and the gene 4 expression weights. And despite this, there is correlation between these QTLs, which induces a TWAS association for gene 4, even though gene 4 is not causal. So how do we get rid of these spurious TWAS results? So previous work has tried to address this question. The Haku et al. Paper suggests post hoc processing, where the idea here is to directly filter out your TWAS results using a colocalization probability of 0.5. So this is not an ideal approach because what ends up happening is you have sort of a binary decision. Is it a PCG? Is it not a PCG? So you sort of end up with a loss of that quantitative information regarding the uncertainty of your classification. And there's also focus, which takes sort of a different approach here. They're trying to model correlation among TWAS signals, essentially trying to identify which gene in a region explains an association signal. But as we'll talk about a little bit later, this sort of approach makes some strong assumptions about the underlying model. Okay, so done with background. Now we'll go with our methods. So our research question to just sort of motivate us here is how exactly can we integrate these two types of analyses, TWAS and colocalization, to implicate PCGs and biological pathways. Okay, so we'll start with a structural equation model to sort of motivate us. So the top equation represents the gene expression for some gene of interest. So that's E, and then we have it as a function of your genotype matrix G and your true eQTL effects beta E. And then the equation below it is for your complex trait Y. So you have that as a function of your gene expression. But we also include this extra G beta Y term, which essentially allows for horizontal pleiotropic effects. Okay, so we're not the first group to essentially try to tackle this problem of implicating PCGs. Others have done this in the past, but their methods often involve additional assumptions. So for example, if you're at all familiar with the Mendelian randomization literature, so usually the goal of an MR analysis is to essentially estimate the effect of your exposure, in this case, gene expression on Y. But some of these methods, for example, MR-Ager regression imposes what's called the inside assumption, and then focus, which we already talked about, assumes that the pleiotropic effects are constant across variants. So these are fairly strong assumptions in our opinion. So we want to get around these. And the way that we do that is focusing more on inferring whether your expression to Y effect is 0 rather than trying to estimate it. So as sort of a springboard jumping off point for our method, we're going to first observe that the causal EQTLs must be colocalized with the GWAS hits based on our SEM. Okay, so sort of taking that observation and running with it, we're going to try to implicate PCGs, incorporating that observation into a formal Bayesian framework, where essentially we are forming a prior based on that colocalization evidence. And we're calling our method integration of TWAS and colocalization, or INTACT. Okay, so getting into some more details about what exactly our method does. So first, we're forming a Bayes factor from our TWAS z-score, and this is representing a marginal likelihood of the SEM. Then we're forming that prior based on the colocalization data. So this is the actual form of the prior, where the pi is an unconstrained TWAS prior. We're sort of adapting an estimator from the Q-value method for this. And then we have f of p coloc, where the f function satisfies two properties. The first of which being that it's a monotonic function of the colocalization data. And the second is that if the colocalization probability falls below some threshold t, then we set it to 0. And the rationale behind this is essentially we want to filter out those spurious TWAS associations caused by LD contamination. So at the end of the day, we can compute a putative probability of putative causality just using Bayes' rule. So one question you might have is, what exactly do those f functions look like? So these are a few examples that are incorporated into our software. So on the left, you have the step function, which is sort of analogous to the post hoc processing that we talked about, where you just filter out any TWAS results that have colocalization probability of less than a half. Right to the right of that, you have the linear function, which we sort of think is the most intuitive option. So this is the default in our software. This is just the identity mapping and truncating a t-value of 0.05. And then the two functions to the right, xbit and hybrid linear xbit, sort of the rationale behind these is that you shrink your colocalization probability if it's less than 0.5, but don't set it to 0. Okay, so we just talked about INTACT in the context of a Bayesian framework, but we've also interpreted it in the context of Dempster-Shafer theory, or DS theory for short. So if you're not familiar, DS theory is essentially a framework for decision-making that generalizes Bayesian reasoning. Basically, our decision here is to decide whether a gene is a PCG or not, and we're trying to integrate two sources of evidence, TWAS and colocalization analysis. And then so what DS theory does is it allows us to quantify the belief towards some decision based on each type of evidence as some mass function. We have two mass functions here, each ranging from 0 to 1. So why we're really interested in interpreting INTACT in this context is for what's called Dempster's rule combination, which allows us to integrate DS-independent sources of evidence. So what does DS-independent mean? So two sources of evidence are DS-independent if they have independent reliability for reaching the same decision. So sort of following our earlier conversation and the background about the different set of genes that can be implicated by TWAS versus colocalization, we're going to argue that colocalization and TWAS are approximately DS-independent. We can't say that they're statistically independent because they often rely on the same GWAS and EQTL data sets. But if we make this argument, we can apply Dempster's rule combination, resulting in this statement here that the overall mass, the overall degree of belief that a gene is causal, given the TWAS and colocalization data, is proportional to the product of the individual mass functions. Okay, so now we're going to sort of more explicitly tie that DS theory formulation to the Bayesian framework. So just a little bit of notation here. We're going to call it the TWAS posterior probability piece of TWAS. So this is going to be proportional to our unconstrained prior times the Bayes factor. So we're going to define our marginal mass functions as essentially being proportional to some monotonic function f or g of your posterior probability for colocalization or TWAS. Apply Denser's rule combination, we can arrive at this conclusion, where essentially the punchline here is that if G is the identity mapping, then we find that the overall mass function is actually proportional to the posterior probability, which essentially means that for our non-model based DS theory framework and our Bayesian framework that was based on our SEM, the overall rankings of genes will be the same based on these two different approaches. Okay, so sort of touching home base here. So what did we just go over? So our goal was to estimate a posterior probability of putative causality for each gene in our data set. We did this using a Bayesian framework where we basically converted a TWAS score to a Bayes factor. And then we designed a prior based on our colocalization evidence that was designed to filter out LD hitchhiking signals. And then we finally interpret it in the context of DS theory. So by this point, you can just think if you're doing the analysis, you have essentially a transcriptome-wide set of posterior probabilities. So the next question is sort of, how do we interpret these more in a biological context? So if we come back to our original research question, you might remember that our goal is not only to implicate PCGs, but also to implicate biological pathways. So again, we're going to be using that posterior set of posterior probabilities basically to find out which pathways are biologically relevant. Okay, so the main way that researchers have typically gone about doing this is what's called a gene set enrichment analysis. So this aims to identify groups of genes that are enriched given gene trait association evidence. So typically this is in the context of a differential expression analysis. So usually input, you'll have your summary statistics from the DE analysis, and then you'll also have a list of gene set annotations. These basically just denote whether or not a gene is a member of whatever pathway you're interested in. You can get these from a few different resources. One might be Gene Ontology or Go, and another might be KEGG. And then for output, you'll often have just enrichment estimates for whatever gene set you're interested in. Some examples of these types of methods are BAGZ and GSEA. So basically what we're going to do is take the BAGZ framework and sort of adapt it from working with differential expression data to working with our posterior probability output. So the idea of BAGZ really is to treat this gamma as latent and then use an EM algorithm to estimate the enrichment parameters. So just sort of as setup to that, we're going to define some notation. So DI is going to be an indicator variable for whether or not a gene is in a gene set of interest. Just to prior here to connect the posterior probabilities to those gene set annotations. So the parameter that we're really interested in here is this log odds ratio alpha 1, where essentially alpha 1, if alpha 1 is greater than 0, then genes in the gene set are more likely to be prioritized. Okay, so more building up here, we're going to form a consistent exchangeable prior using the strong law of large numbers and a total expectation law. And then we're going to use that to formulate a Bayes factor for each gene as the ratio of the posterior and prior odds. And then finally, getting into our EM algorithm using those gene level Bayes factors. After initializing our alpha vector, we're going to first update the conditional expectation of gamma in our E step, and then our M step performs a 2x2 contingency table maximization to update the alpha vector. So we iterate between those two steps until we get convergence. So in practice, what we find is that when the underlying true contingency table is highly unbalanced, that could be if basically one of your cell counts is very close to 0. Basically, we have problems with the algorithm converging. So in order to account for that, we shrink the alpha 1 estimate by imposing a standard normal prior. And then finally, for uncertainty computation, we use a numerical differentiation algorithm to compute standard error of alpha-1. Okay, so just coming back to the sort of the very high level view of both INTACT and INTACT-GSE. You've already seen the sort of upper left portion of what's going on. That represented the INTACT procedure, and then what we've just gone over was essentially using the posterior probabilities from that INTACT step, and in addition to the pathway annotations to perform gene set enrichment estimation using INTACT-GSE. Okay, so done with methods. Now we'll go on to a simulation study to see how this works. So here's some of the details of the simulation study design. So we're using genotype data from 500 individuals from the GTEx project. We're randomly selecting about 1,200 genes, each with about 1,500 common cis-SNPs. We're selecting 2 true EQTLs per gene, and we're annotating each gene as either or not a member of a gene set with essentially probability 0.4. Then we're assigning a causality indicator for each gene based on our logistic prior and the gene set annotation. And finally, we simulate both gene expression and the complex trait, essentially based on our structural equation model that I showed before. We're drawing the residual error variances from a standard normal. Each of the effect sizes are being drawn from a 0, phi squared, or in this case, phi is 0.6, because that's essentially the value that we found most accurately reflects observed signal to noise ratios. And then we're simulating 100 data sets for several different alpha-1 values for a total of 500 data sets in total. So after doing all this for each data set, we apply statistical fine mapping so that we can do colocalization and TWAS. So getting into those results, so essentially what we're doing here is comparing the performance of TWAS only methods, colocalization methods, our post hoc processing, and then finally INTACT. So the way this table is set up is you have the realized false discovery rate at a target control level of 5% as the first row, and then the second row you have power. And what I want to draw your attention to is over here for the TWAS only methods, you see that they have the highest power, but they have excessive false positives. You see the FDR is not actually controlled. The colocalization methods here in the middle, you can see that they're generally relatively conservative compared to the other methods. And then finally, if you look all the way over here on the right, INTACT using the linear prior, you can see that this actually has the highest power of all of the properly FDR controlled methods. Okay, so that was INTACT. Now we're going to look at how INTACT-GSE performed. So the way that this figure is set up is you have the true enrichment parameter alpha-1. So there are 5 different ones of these and 100 different data sets for each one. And then on the y-axis, you have the estimated enrichment parameter. So the dot in the middle of each of these is the mean enrichment estimate, and then you have the corresponding 95% confidence interval. So the red is INTACT-GSE, and then we're comparing it to a 2-stage approach, which essentially only uses TWAS data. So the first stage of the 2-stage approach is to essentially classify a gene as putatively causal or not based on a 5% FDR threshold. And then using those binary classifications, the second stage essentially performs a 2x2 contingency table maximization. You could basically just think of it as a logistic regression of the indicators on the gene set annotation indicators, the DIs. So there are a couple of things I want to point out here. First of all, you can see the mean enrichment estimate for INTACT-GSE is less biased generally than the 2-stage approach. And we think that's because we're incorporating the uncertainty from the INTACT output, whereas the 2-stage does not. And also you can see that both approaches are downward biased, and a couple of reasons we think for that are, first of all, that you have classification error. You saw the power for basically none of the methods have perfect power. also the fact that the INTACT-GSE algorithm involves the shrinkage step. So that might be another cause. Okay, so that was our simulation results. So now we'll move into some real data application. So for the real data application, we're going to apply INTACT to 4 different UK Biobank molecular trait GWAS, as well as the multi-tissue GTEx EQTL data. So we're looking at serum urate, IGF-1, and then testosterone levels separately for males and females. So why are these traits of particular interest to us? So these are actually pretty well understood at the molecular level. So the sort of the rationale here is we can use these to benchmark, are these methods implicating the right genes or are they not? Okay, so the way that this table is set up is each of the entries correspond to a method and a trait. And essentially it represents the number of gene tissue pairs that are implicated by the method. And then the number in parentheses by the side, to the side of it is the total number of unique genes across tissues. So there are a lot of numbers here, and basically the only thing I want to draw your attention to for now is the fact that you can sort of see the same pattern as we saw for the simulation. In which, if you look at the total number of gene tissue pairs implicated for the default linear prior, that falls between the fast and lock colocalization approach as well as the TWAS approach. Okay, so next we're going to basically compare INTACT to the method that we sort of touched on in one of the first slides, the first Manhattan plot that we looked at. So if you remember, I sort of talked about that proximity plus knowledge-based approach, which involves linking GWAS hits based on previous biological knowledge and genic distance. So here what we have are the 4 different traits in the first column. And the next column, we have the number of GWAS loci based on this paper. The next column is the number of PCGs that we're implicating with INTACT. And then to the right of that, we have the number of core genes that are annotated. So these core genes, this is terminology from the omnigenic model, but basically you can just think of this as, think of these as genes that are key players in the underlying biology of the trait. So basically the big takeaway from this table is you can see that since INTACT is not sort of limited by the biological knowledge, that sort of predefined list of genes, you can see it implicates a lot more than the proximity plus knowledge-based approach. Okay, so now we're going to look at the performance of INTACT in implicating what we call those core genes in comparison to the proximity plus knowledge-based approach. So now we're sort of further breaking down each trait by subpathways. So you see that the pathway column gives the name of the subpathway, and beside each name, you can see that there is the total number of core genes in parentheses for that given subpathway. So the column to the right of this is the number of core genes implicated by the proximity plus knowledge-based approach. To the right of that is the number implicated by INTACT, and then to the right of that is the number implicated by the overlap between the 2 different approaches. So if you sort of scan this table, at least in comparing this column here to this column here, you can sort of note that the proximity plus knowledge-based approach appears to be implicating more core genes than the INTACT approach. But there are still cases where INTACT is implicating more core genes that the proximity plus knowledge-based approach is not. So there's a few different reasons that we've come up with to try to explain this discrepancy. The first, it's possible that for the core genes that we're looking at in a particular pathway, remember we're implicating transcriptome data using INTACT, and the transcriptome might not necessarily be the most relevant particular omics platform for the given core gene. And in addition, the proximity plus knowledge-based approach does not incorporate an explicit FDR control mechanism. So that could be another reason for this discrepancy that we're seeing here. Okay, so finally, we're going to look at how INTACT-GSE performs in implicating these 4 gene subpathways in relevant tissues. So we're basically getting these relevant tissues from previous biological knowledge that's reported in this paper. So the way this table is set up is we have the trait all the way on the left, to subpathway again to the right of that, and then each entry corresponds to a tissue-specific enrichment estimate, the alpha 1, and the corresponding standard error. So the statistically significant estimates are in italics. There's 9 of them of the 39 tested tissue pathway trait combinations. And then the NI entries mean that essentially the observed data is non-informative. So you can just think of this as cases where the INTACT-GSE EM algorithm did not converge. So there's a few different explanations we've come up with this, possible explanations we've come up with for this too. One of them being that, again, that gene expression could not be the most relevant molecular phenotype. And also some of the sample sizes for the tissue-specific GTEx samples were sort of on the small side. For example, I think the kidney cortex sample size was about 70 or so. All right, so just a little bit of wrapping up conclusion here. So we saw that INTACT identified genes with higher power than colocalization analysis and properly controlled FDR, properly controlled the type 1 errors that might have been induced by LD hitchhiking. And then for INTACT-GSE, we saw that this was relatively accurate compared to that 2-stage approach. And that's probably because it leveraged the intact output for the uncertainty quantification. So there are definitely limitations to this approach, some of which we've sort of already seen. First of all, we're only considering one molecular phenotype at a time, just expression for now. We're actively working on an approach that can incorporate multiple molecular phenotypes. So we've seen in previous work that the available data sets that we have for colocalization analysis have actually poor power for uncovering colocalization instances. So, you know, this is an issue, right? If you're using your colocalization data as a prior. Next, we can't identify trait relevant tissues or cell types. We needed basically previous biological knowledge to point us to those. And we can't estimate gene to trait effects using this method. It's really just designed for inference rather than estimation. Okay, so if you're interested in this work, this is the corresponding manuscript. And we also have an R package on my GitHub, working on getting it onto Bioconductor. And finally, I'll give a little bit of I'll talk a little bit about the R package. So basically, if you go on my GitHub and just look at the README file, you'll see this top bit here with examples. So there's basically 2 functions here that you'll be interested in, INTACT and INTACT-GSE. So I've included some sort of toy data to play with just to show how it works. So there's the object SIMDAT, which basically includes your genes and then corresponding colocalization probabilities and TWAS Z-scores. So if you apply this INTACT function here, this will essentially spit out a vector of the same length as the GLCP and Z-scores, just with your posterior probabilities. And then there's also this INTACT-GSE function where basically if you plug in your data frame and your gene set list, which is formatted like this, is just a list object with vectors corresponding to genes that are member genes. Basically, it'll spit out a data frame where you have your alpha 1 estimate and standard error for each gene set, and you also have basically whether or not it converged or not. So I'd like to thank my advisor, William Wen, for help with this work, as well as our co-authors, and I'd be happy to take any questions.
Anyone online has a question? Again, you can either put that in the chat box or raise your Zoom hand in the reactions on the bottom right. And if anyone in the room has questions, feel free to raise your hand and ask.
I guess when doing those colocalization studies, do you, like, what's like the range of the EQTL and like versus like the variance that you're testing in that range, or is it like a very precise variance at that point?
The range of the, so do you mean like how close they have to
Yeah.
be? I think generally so the signal clusters are generally in cis to the gene, and I think generally the threshold for that is like 1 megabase on either side of the TSS. Yeah, good question.
I have a question. You may have mentioned this, but why did you choose to use BAGSI instead of GSEA?
So the way that BAGSI was formulated is it basically made it sort of the only difference was the way in which we actually incorporated our posterior probabilities versus the output of a typical differential expression analysis. And it's sort of naturally set up as an empirical Bayesian framework that just made it easier essentially to use BAGSI than GSEA. Good question.
Nothing online yet, but we can give it a couple minutes in case anyone's typing, and if you got anyone else in the room has questions, feel free to ask. We do have a question online, so
Okay.
Dana just wrote, great talk and interesting method. Can you give us a sense of the scale of data available for TWAS, especially compared to available GWAS data?
Yeah, so there are definitely a lot of TWAS data sets out there. I believe methods like, S-Prediscan and the TWAS method itself are designed to work with GWAS summary statistics. So it's getting easier and easier to just do a TWAS using GWAS. Yeah, I don't know if that helps. And there are a lot of, I believe, a lot of authors post their summary statistics for TWAS in particular. So I think that there is it's definitely not to the point that the available GWAS data sets are, but there's definitely a growing set of data out there to do these types of analysis on.
Dana said yes, that was.
Okay.
Again, maybe just give it another minute or two in case anyone online is typing.
Sure.
Well, not seeing or hearing any other questions, so let's thank Jeff one more time. Thanks everyone for coming, and hopefully see you next week.